Let's try to understand desensitization process in the GPCR signaling. How this happened, why this is happening, and we will understand everything in this video. In the desensitization process, why we are doing the desensitization, we want to stop some sort of the stimulation because it is too excessive in our body. So what we can do, we can remove completely the receptor from the plasma membrane, or we can do the recycling process so that we will get some time to recover our cell. So what really happening here, you can see there is a GPCR receptor is present on the plasma membrane, which is made up of the protein, and it have N and C terminal, as you can see. And also it is crossing the 7 times to the plasma membrane. Whenever the ligand will come, any ligand is binding on this GPCR receptor, and this GPCR receptor is bound with the 3 subunit trimer condition, alpha, beta, and gamma. Whenever the ligand will come and bind on the receptor, what will happen? GIF activity will get activated. So this GIF will add the GTP on the alpha subunit. So what will happen? This alpha will move towards the adenylate cyclase. Only alpha will move here, and alpha will responsible for the activation of the adenylate cyclase because of the response came from the ligand here. And what it will do? It will make the camp with the help of ATP. So ATP is required to make the camp. After formation of the camp, this will form the pKa. So what pKa will do? It will activate the Krebs with the binding. So Krebs will get activated. What does mean by Krebs? You can say the camp response element binding protein. So the pKa is activating through the phosphorylation process. You can see the Krebs get phosphate on it. So the Krebs get activated. This Krebs will go and bind with the CBP. This Krebs and CBP together will go and activate, means transcribe the one particular gene, which is known as a GRK. This gene get transcribed and RNA will form. This RNA also called as a GRK RNA. After formation of the protein, we are calling it as a GRK protein. This GRK protein is nothing but G protein coupled receptor kinase. As we know, the kinase is nothing but the phosphorylation is happening in this process. So it can do this, protein can do the phosphorylation on anything. So what it will do, it will use the ATP and remove the ADP, and it is also working as a feedback inhibition process. Because why this is calling as a feedback? Because as we know, this GPCR receptor is we want to remove this GPCR. So when the excessive amount of the signaling came from this side, means from the ligand, so what will happen? We need to give some feedback. Don't do any excessive processing system or any synthesis process inside the cell, because excessive also not good for the body. So this is the feedback inhibition process. So our GKA will do phosphorylation on the GPCR receptor. After phosphorylation of the GPCR receptor, arrestin protein will come and bind on the GPCR receptor. After GPCR get arrestin protein, the clathrin will come and make vessel. And because of the formation of vessel, what will happen? The endocytosis process is going to happen, and we will take the GPCR receptor inside the cell. So after it will come inside the cell, it will enter into the early endosome. From here, what can happen? Either it will go towards the lysosome and degraded in the lysosome, or what we can do the recycle process. You can see here. In based on what we are doing the recycle and lysosome, if the receptors are too much high on the plasma membrane, we will send these GPCR into the lysosome. But there are some receptors only present, but we want to send back because there is not that many receptors are present on the plasma membrane. So what we can do the recycle process. And why we have taken here inside the GPCR, again we are sending back to the plasma membrane, because in between this all process it will take lots of time. So what will happen? The stimulation process gonna be reduced, and the signaling amount also reduced. So our cells will be do proper function. If you like this video, please make sure to subscribe.